And so after we make our protein, where that occurs is on the ribosome. And remember from when we talked about ribosomes in the cell chapter that they can be attached to the rough ER or be free floating around in the cytoplasm. And so that's what this is, the free and the bound. So free is, free means that they're the ones floating around in the cytoplasm. You can write that here. In cytoplasm. Bound is attached. They could be attached to the rough ER, RER, or they can also be found in the nucleus as well. And so they do the same in that they make polypeptides. And then usually the ones attached to the ER, the making of the polypeptide continues, and then they're exported from the cell. So if you remember the rough ER, a vesicle buds and has the protein attached to it. This vesicle goes and binds to the Golgi apparatus, where the Golgi apparatus modifies the protein some more. Buds off, the vesicle buds off the Golgi apparatus and binds to the cell membrane. Do you guys remember that a little bit? That's what's called the endomembrane system. So it's pulling in a long time ago. So this is a picture of a bound ribosome. And so this ribosome right now is a free ribosome. It's not attached to anything beginning. This is your mRNA strand beginning the polypeptide synthesis. Here's bound ribosome, polypeptide ends up in the ER. So this is the inside of the ER, and then a vesicle can bud off of there and go to the Golgi apparatus where it can be further modified. So once in the lumen of the ER is the inside of the ER, the newly synthesized peptide is modified. The protein then undergoes further processing, I'm going to put in parentheses here, in the Golgi apparatus. And then is shipped where it needs to go, so whether it be another organelle or to the cell membrane, and so on. So, RNA, and we learned about 3 kinds of RNA, mRNA, tRNA, and rRNA. These are 3 things that are helpful in all kinds of RNA. They can hydrogen bond to other nucleic acids, can form a 3D shape by hydrogen bonding on itself, and can act as a catalyst. So number 1 here, it can hydrogen bond to other nucleic acids. So if this is your RNA with the sugar phosphate backbone, and you have A's, C's, U's, and G's hanging off of it, so this is one like an mRNA or a tRNA. Since it's single-stranded, then they can form hydrogen bonds. So if there's a base with a U, this A can bind to it because it doesn't have another side to it, and so it can hydrogen bond with 2 other nucleic acids. It can form a 3-dimensional shape that's like tRNA. So tRNA, when it's made, it's in a straight line, but then it folds on itself and has that 3-dimensional structure, folds on itself and so on, where it has the amino acid attachment site at one side and the anticodon on the other side. And so we drew that in our book. They say it like this, where this is the tRNA and here's your anticodon, and then this is the amino acid binding site, but it has this 3-dimensional shape. So that's what that means. So when making proteins in bacteria, prokaryotic cells versus eukaryotic cells, there's some differences between them. Prokaryotic and eukaryotic RNA polymerases are different. So the enzymes are a little bit different, but they function the same way in that they both, RNA polymerase makes mRNA. Transcription also is terminated a little bit differently in bacteria than in eukaryotic cells. Prokaryotic and eukaryotic ribosomes are a little bit different in the small and large subunits. Transcription and translation are streamlined in prokaryotes and is compartmentalized in eukaryotes. And eukaryotic cells have a complex system of targeting proteins for their final destination, like are they going to go through the cell membrane and so on. What does it mean when we say that they're streamlined in prokaryotes? In prokaryotes, transcription and translation both occur. They both occur in the cytoplasm. That is not the case when I say both. That's transcription and translation. They both occur in the cytoplasm. In eukaryotic cells, it's compartmentalized. So where does transcription occur in eukaryotes? In the nucleus, because that's where your DNA is. So transcription is the nucleus. And where does translation occur? Is at the ribosome, which is out in the cytoplasm. So they have two separate locations. That's what it means by compartmentalized. So the last thing that we have to talk about is how a mutation, which we've talked about mutations before, what a mutation is, but how in the world does it affect protein synthesis? So in looking at this, mutations are changes in genetic material, so we know that. And they can cover large portions of DNA or can be a point mutation or affect one single base. That's what that means, at one particular point. So we're going to look at two categories of what we call point mutations. Base pair substitutions, and that's just like it sounds like. So if you have an A in the DNA, we take the A and substitute it for another, like so for a C or something like that. So we substitute a base. Or insertions and deletions, which is exactly how it sounds, inserting an extra base or deleting a base. And so what kinds of things can cause these things to happen? And so an error in replication. So when every time our cells divide and we replicate our DNA polymerase adds the appropriate bases, so we have two identical pieces of DNA. Sometimes that can result in the wrong base being added. Most of the time there's enzymes to repair that, but if it doesn't work, then we end up with a mutation. Or a class of outside substances called mutagens that can cause your DNA to mutate. So we've talked about UV light being able to do that, certain chemicals that can cause mutations. So this picture here shows you the wild type and the mutant DNA for the protein hemoglobin. So hemoglobin is the protein that's found in our red blood cells that is useful to carry oxygen from our lungs to our body cells. So here's the piece of DNA. So this is DNA, this is the wild type, this is normal. And so here are your bases, and they've only highlighted these three bases here. They didn't put all the bases on here. The actual gene would be much longer because this is the part that we're going to see is mutated in people who have sickle cell disease. So when we go from DNA to mRNA, this process is transcription. So what goes with the C is a G, A, and A. And notice here, DNA is normally double-stranded, right? But we unzip and break the hydrogen bonds and only use one side of the DNA as the template. And the mRNA has to grow in the 5' to 3' direction, so the DNA is from the 3' to the 5'. So therefore we have our codon here, GAA. And then it goes to the ribosome where translation occurs, where we read, quote unquote, read the mRNA in groups of 3 called codons, that each one stands for an amino acid. So remember, so we have 3 codons here. This codon, they don't have anything on there. This stands for a particular amino acid, but it's part of the hemoglobin molecule, but isn't part of what's going to affect and be mutated. And so we have GAA. If you look in your chart and look in GAA, you will see GLU. All right, it's glutamine. And so this amino acid is supposed to be in this particular spot to make a normal hemoglobin. Remember, your amino acids are really important in order when your protein folds the R groups to interact and give you the right shape. And so the shape is really important for the function, which is carrying oxygen. So people with sickle cell disease, their whole gene for sickle cell disease is right except this one base. And so instead of CTT, it has CAT, so that T is substituted for an A. So that's a single substitution, which then, when it's read in groups of 3 here, affects this codon here. So it has a G, and then instead of an A, we have a U that goes with this, and then an A. And so if we look at here, GUA, if you looked in the chart and looked in GUA, it stands for a different amino acid. It stands for valine. And so what we have here is a change in the amino acid, which this amino acid happens to be instrumental. It's one of those amino acids that the R group is important in bonding with another R group to give the hemoglobin its right shape. And so this amino acid is important. So then if you change that amino acid and give it to valine, then the R groups don't attach and they get a different shape, which affects its function. People have sickle cell disease here. So that's a single substitution. Let me ask you this. Is it possible, do you think, to have a mutation in your DNA but not affect your protein?
Yeah.
Yeah, absolutely. Can somebody give me a scenario in which that would happen? Can you give me an example of how that could happen? Yes? That's correct. So remember that third base, I used the example on Friday, like CCC, CCG, CCA, and CCU. These are all mRNA codons that code for the amino acid proline. So if you had a DNA strand that had GGG as its codon, then the mRNA that would be made here would be CCC, and the amino acid would be proline. And so what would happen is if I mutated this, if the DNA mutated and this G is now a C, that's going to then change the codon in the mRNA to CCG, but CCG is still proline and it doesn't change my amino acid.
Also, aren't there just some genes that never get or is there just too tightly coiled?
That's right. So if you had a mutation in an intron or you had a mutation in a gene that is encoding for making a particular protein, then right, you wouldn't see that. And so, absolutely. This actually, my niece she had a bunch of genetic testing done when she was a baby because she had some symptoms. They thought maybe she had some kind of genetic disorder, and they did some genetic testing and found a mutation on one of the DNA, a specific gene in her, in a piece of DNA in one of her chromosomes. And they thought maybe they found that might be causing her symptoms. And then they tested my sister and her husband, her parents, and found out my brother-in-law, the baby's baby's dad actually had the same mutation and he had no symptoms. So then they ruled that out, and the geneticist told them that we have gotten so sophisticated in our DNA testing that we actually find mutations, but the mutation doesn't affect the overall protein that's made and therefore the function of it. And so they're finding more and more where people, when they do genetic testing, finding more mutations, but they necessarily aren't causing the symptoms that are. So that would be an example of a silent mutation, right? So, so base pair substitution, this is called I use the word silent mutation. One base pair gets substituted for another, that's what we just looked at. Sometimes these are silent, that's the example I just used, because of the redundancy of the genetic code. So oftentimes in that third base, if that third base is mutated, it oftentimes doesn't change the amino acid. Other times they're silent because the mutation codes for an amino acid or protein that's not essential to the protein's function. Also, there are times when the two amino acids behave the same even though they are different. So, for example, other times they are silent because the mutation codes for an amino acid or protein that's not essential to the protein's function. So, only certain, remember I said that if my arms, I'm the chain of amino acids, and so you have hydrogen bonds or R groups interacting to give it a particular shape. What happens is some amino acids aren't involved, the R groups aren't involved in the bonding and therefore the shape. So if you have an amino acid that has been replaced with another amino acid and it's not essential in causing the shape of the protein, it may go unnoticed as well, all right, and it'll cause the shape change. And then this one here says the two amino acids behave the same even though they're different. So you can have two amino acids and their R groups be different, but their R groups still behave the same way. So for instance, let's say you have an R group here and an R group here, and they hydrogen bond to each other, and that helps with the shape of the protein. let's say I change this amino acid to a different amino acid, but its R group still hydrogen bonds to this, then you still have that hydrogen bond there, and therefore the shape of the protein stays the same. And so that's an example. So then you wouldn't have a change in phenotype or physical appearance as a result of that. So this goes and shows you 3 different examples of types of substitutions. Let me show you here what we're looking at. So at the top, this is your wild type. This is the mRNA. So not showing you the DNA, so they're just showing you the mRNA. And here are your codons. And if you read these codons in groups of 3 and use the chart to do that, this is the order of amino acids you would have. So AUG is the starter methionine, and so on and so forth, until you get to UAA, which is the stop codon, and that doesn't stand for an amino acid, so therefore its last amino acid is here. Okay, so that's the wild type, and this is the protein you're supposed to make. So this one shows you a substitution in, these are the codons. This codon here, second to the last codon, instead of, it's supposed to be GGC, it changed and put a U instead of a C. That's why it's a substitution. We're substituting one base pair for another. If you use that chart, notice this is the third base, just like this, the change in that third base oftentimes codes for the same amino acids. So you have the change in that third base. If you use the chart GGU, it actually stands for glycine. Glycine is the same amino acid as that GGC stands for, and so therefore this is a silent mutation. Because, do we have a mutation? Absolutely. We do have a mutation, but it didn't change the overall protein. In this one here, we have AGC here instead of GGC. So now we're changing and substituting the G for the A at the first base in this codon. And then if you use the chart AGC, that does stand for a different amino acid. So we have SCR here, it's supposed to be GLY, and so we have a change in the amino acid. Could this affect the protein? Absolutely. Okay, so that could affect the protein. This says, this missense, that means the mutation changes the amino acid.
Sense.
And then here, same thing, we have a substitution over here in the second codon. The second codon is supposed to be AAG, which stands for lysine. Now the A got converted to a U, substituted with a U, and then that's one of our stop codon. So here, does it even matter about these other codons? No, because it's going to stop and never add those amino acids. So this is a really bad case scenario because it's not made hardly any of the protein whatsoever. And so therefore, that individual, if that mutation occurred in you, you wouldn't make that any protein, basically any of that protein. And so all of these 3 examples are all examples of substitution. So example 1, 2, and 3, where we've substituted one base for another. And they can cause different things to happen. So sometimes it's no big deal, other times it is. So these couple bullets are about those. So missense mutations code for an amino acid, but usually the wrong amino acid. So it changes the amino acid. And then in a nonsense mutation, the wrong amino acid is encoded and it usually results in a stop codon, and then you get a non-functional protein. Which, going back up to this picture here, we could write, I wrote here it changes the amino acid. This, we can write changes to a stop codon. The last thing that we need to look at is insertions and deletions. So these 3 were substitutions, substituting a base for another base, and so insertions and deletions. These are often referred to as what we call frameshift mutations because they alter the reading frame. What do I mean by that? When we read mRNA, we're reading them in groups of 3 called codons. So the reading frame is the codons we're looking at. And remember, the reading them in groups of 3 is really important to bring and get your right order of amino acids. And these types of mutations are always, you get a non-functional protein, basically. Let me show you why that is. So this is our last picture here. So this picture looks very similar to the last picture. You have your wild type and your mRNA and the protein that you're supposed to make, except now our examples are with insertion and deletion. So we have 3 examples of this where we insert an extra base or take out a base. And so, so notice here, the first codon is supposed to be AUG, so that's the same, AUG. The second codon is supposed to be AAG. Now notice in this picture, AAG is still there. There's the AAG, but look at, they added or inserted an extra U here. So if they put a U here, then that shifts everything to the right, right? That's why they call the frame shift. So now if you add in a U, then the next codon, instead of being AAG, now is UAA. And that G for this codon becomes the first base of the next codon. So then the UUU is still there, but it's not the next codon because it's going to be GUU, all right, and so on and so forth. So it's all shifted to the right in this case here. So in this one, this happens to be, inserting this extra U is really detrimental because it makes the next codon UAA, which if you look in your chart is a stop codon. And so therefore, that's why it says causing immediate nonsense, that's stop. So it makes the second codon a stop codon, and so therefore it's really detrimental. You're not going to get that protein made. So it's insertion or deletion. So now this is an example of a deletion. So here you have your codons. The first codon, AUG, isn't affected. The second codon, AAG, isn't affected. The third codon is, notice here it says the U is missing. So in this case here, this is a deletion. So they're taking out a U, so this is a deletion. Taking out the U. So if they take out this third U, the third codon was supposed to be UUU. If you take out that U, then that everything shifts to the left, so this G is going to be the third base in that codon here. So the next codon instead of UUU is going to be UUG. And then the next codon is GCU, all right, GCU, everything shifted over. And so what happens in this case is that when we took out this U, it not only affected this codon, but everyone after that, okay? And so therefore, every amino acid after that will be changed. Did we change our protein pretty good? Absolutely, okay? And so it can be a stop, which changes it. It changes all the amino acids after the mutation. Or in this case here, sometimes the insertion or deletion could be a whole codon. So in this case here, they're showing you it's missing, so this is a deletion, so they're taking it out. So instead of just taking one base, we have AUG, the next codon is supposed to be AAG. They take it out, so the next codon, the second codon is UUU. UUU is supposed to be the third codon here, and it's not. And so in this case here, it doesn't cause your codons to change. The UUU is still together, the GGC is still together, and the UAA, but you're just missing one whole amino acid. So right here, you were supposed to have the amino acid lysine, but you don't have that amino acid because the whole codon was taken out. Question?
In the frameshift, when you go to missing, what happens to the stop codon?
Yeah, so in this case here, the stop codon, now AA and whatever that is, now you don't have a stop codon, so it will continue on until eventually it reaches either the end of the RNA molecule or reaches this stop codon. Yeah, yeah. So you don't get the correct protein. Good question. All right, and that is the end. So I'm going to pull this all together for us. All right, so I'm going to draw some things up on the board to kind of go over the whole big picture of protein synthesis. So my board is going to be part of the cell. So this is the nuclear envelope. Here's the nucleus. Here are the nuclear pores. And out here is going to be the cytoplasm. So we're going to go over this whole process here together. So we're going to do for eukaryotic cells. Things are a little bit different in prokaryotic cells, but we're going to do eukaryotic cells. So it all starts in the nucleus with what one molecule? DNA, right? And so we have, let me draw here, your DNA molecule, double-stranded. And I'm going to just do the basis of our gene. So let's do, this side is going to be TAC, so then it's going to be ATG, then let's do okay, so let's say this is our DNA. And let's put the 5 and the 3 prime. So 3 prime, 5 prime, 5 prime, 3 prime. So it's anti-parallel. So notice I have pieces of DNA hanging out here because I want to show you that your gene is just a segment of that DNA. Okay, so that's where our DNA is. The first step of protein synthesis is called transcription, right? Transcription is making an mRNA, right? What enzyme helps to make mRNA from DNA? Rna Polymerase, specifically too. Rna Polymerase is the enzyme. And so you have this large DNA molecule. Your gene is only a small section of it. My question is, how in the world does RNA polymerase know where the beginning of your gene is? There's a promoter region that's before the gene. And specifically, there's a specific order of bases that are part of the promoter region called the TATA box. All right, so because of the order of bases are TA, TA. All right, so let's write this. So this is going to be, I'm going to write just TA, TA. Notice that they call this upstream. It's before the gene. So then the bases on the other side would look like this. And they call this the TATA box because of the order of the bases, TA, TA. All right, so with that, the TATA box is part of this promoter region. And so you have these molecules called transcription factors that come and bind to it. And then when those transcription factors bind to that TATA box, that helps to know for RNA polymerase to know where to bind. RNA polymerase does a couple of things to make RNA. It untwists the DNA and breaks the hydrogen bonds and also adds the nucleotide. So it kind of does a lot of different things. So I'm going to draw this next picture and I'm going to kind of put a couple of different steps all in one. So I'm going to have here, this part here, and then when I get to the gene, I'm going to have it separate and have a little bubble like this. All right. All right, so this is my DNA, 3, 5, 5, 3. Here's my TATA box upstream for my gene. And then dot, 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 we have bases in between. And then here's the start of my gene, so I'm going to put my first couple codons in here. So this side will be A, T, G, A, G, C. These guys will still be together. So not part of the bubble yet. All right, so this is what we have going on. So let me explain. RNA polymerase did this, where it separated the two sides of DNA. And so how does RNA polymerase know to bind in where that's the start of the gene? And so we have these transcription factors that bind to the TATA box. All right, so this is transcription factors. Part of the promoter region. So you have these transcription factors bind to the TATA box in this promoter region, and that's what helps RNA polymerase So I'm going to draw here this enzyme R. So this is Rna polymerase. So it knows where to bind. RNA polymerase, then that's how the DNA got untwisted and broke the hydrogen bonds because of RNA polymerase. And then RNA polymerase begins to add RNA nucleotides just to one side of the DNA because RNA is single-stranded. So my question to you is, will it use the top strand of the DNA or the bottom strand of the DNA to make its mRNA? Top strand, because let me tell you why. The new strand needs to grow in the 5 to 3 prime direction. So therefore you have to use the top strand in the opposite. Does it make sense? All right. So 5 to 3. And so if I'm RNA polymerase, my first base now is there's a T I come into contact with, so I'm going to add an A. An A, what am I going to add next? A U, because it's RNA. Then a G, then an A, then a G, then a C. Okay, so I'm building my RNA strand here. So this is the phosphate sugar backbone right here. These are the bases sticking out. And so what will happen, that I'm not going to draw lots of pictures to show you this, but this RNA polymerase will continue to move and it'll separate the gene here and continue adding bases until you get to the end of the gene. So that's what RNA polymerase does. So at the end of this, what you get, the DNA will go back together and you'll get an RNA strand like this. So here's your RNA. It'll have, here's the built in the 5 to 3 prime direction. It has AUG, then AGC. What would be the next codon? The next DNA is CCC, so it would be GGG. And the last one would be UAA. So this is we call this our pre-mRNA strand in eukaryotic cells. So this from right here to right here, what we just did is transcription. So then, why do I call this our pre-mRNA? Because what do we have to do to it? We have to send it out to the cytoplasm. We do a couple of things to it before we send it out to the cytoplasm. Yeah, 5 prime cap and poly A tail. So we add a cap on it and a tail here. The purpose of this is called the poly A because it has lots of A's. Cap and tail. The purpose of the cap and tail is, as we said, the mRNA is eventually going to go out into the cytoplasm, and the cap and the tail help to protect it from being broken down by enzymes in the cytoplasm. So that's the first thing we do. One of the things we do, I should say. And then what else do we do in eukaryotic cells? We do splicing, which is cutting out the introns. So let's say from here to here, the beginning to there is an exon. Let's say this is an intron, and this is an exon here. So when we do our RNA splicing, so this is our pre-mRNA strand, we are going to cut out the introns. And so since this is an intron, we have a molecule called a spliceosome that kind of cuts the intron out. And so then we end up with our final mRNA. So this is your final mRNA. And the order of bases would be A, U, G, A, G, C. We cut this out, so now we have U, A, A. And we have the 5 to the 3 prime. All right? And this is our final mRNA molecule. This part right here, from here to here, we call RNA processing. And does this happen in bacteria? No. This is in eukaryotes only. Now let me redraw here's the cap and the tail here. on there. Now what happens to our mRNA? Now it goes out to the cytoplasm. So it's going to go out through one of these nuclear pores, and where in the world is it going to go?
Be free. Bye.
It's free, it's floating around, it's going to bind to something. The ribosome at this point, what does it look like? Two parts, right? So you have what's called the large subunit, and you have the small subunit. Which one does it go to? Small, right? So here comes your RNA. It's going to come over here, and here it is. All right, so this is your mRNA, 5 prime, 3 prime, cap and tail. All right, it made it. It didn't get broken down in the cytoplasm, and here it is at the ribosome. Now let's look at the first codon. The first codon is kind of like in the middle here of the small subunit, AUG. So before the large subunit binds to that, what happens is the first amino acid is brought. How do I figure out what my first amino acid is? I use my mRNA codon, AUG, go to my chart, and that stands for methionine. So you have a bunch of amino acids floating around in your cytoplasm, and one of those is methionine floating around. And so this is floating around because you've eaten protein, and so you've digested protein and had it in your cells. So your amino acids floating around. Now how am I going to get methionine to my first codon? What takes it there? tRNA. So now I have also, our cells make a bunch of tRNA. So you have one tRNA. What makes them one different than another is the anticodon that it has. So we have a bunch of different tRNAs with a bunch of different codons on it, like this, and so on and so forth. So, we make all these different tRNA molecules. So what happens is, notice here, AUG, the only tRNA that will bind to that is the tRNA with UAC, right? So it's UAC. And so this is the only one that will bind to that. No other tRNA will bind to AUG. The thing is, why when we look in our chart the AUG stands for methionine is because this particular tRNA, that's the only one that can bind to this particular tRNA can only match up and bind with methionine. So out of all the 20 amino acids, this tRNA can only bind with methionine, and so therefore that's why AUG stands for methionine, because the only tRNA that can come and bind to that is the tRNA that has methionine on it. Make sense? And so this little guy here comes over here and binds it. So we have our first amino acid. Now in comes our large subunit. So now our large subunit binds. And remember that there are 3 binding sites, EPA. Our first amino acid is in that middle binding site, which means our next codon, which over here, our next codon is AGC. So our next codon is now in the spot for the A site, and the third codon is kind of hanging out over here. All right, and so this codon right here, the next codon AGC, is in that third binding site, which then means that another tRNA molecule can come and bind and bring the second amino acid. So what I'd like you to do is in your picture to draw the tRNA molecule in that third site, draw the anticodon, and what amino acid would be attached to that tRNA molecule. So you're doing that for the next codon, AGC. So what would the tRNA look like in that site? What amino acid would it be with? All right, so
Hi, Auntie. Will you freeze for a minute?
downtime I'm just passing out.
Oh no.
I'll go over this.
Hide that.
All right, I'll finish it. All right, let's look at what you should have here. So your anticodon should be UCG, and the amino acid. What are you using in the chart? The mRNA. So AGC was what you should have looked in the chart, and what did you get? SER, S-E-R. Right, that should be your second amino acid. And so now we have our first amino acid and our second amino acid. Now what do we want to happen between those amino acids? We want them to bind together. So what happens is, then I'm going to pick a different color to show you here. This bond breaks, and a new bond is formed between your two amino acids. So the X stands for the breaking of the bond. So methionine MET is no longer attached to this tRNA, and is now on this tRNA. So this tRNA has two amino acids, the first and the second, attached to it. And so now we have our first two amino acids. Now we want to bring the next amino acid, so we want to read the next codon. But notice the next codon is hanging off. So what happens? Everything shifts over one codon. So let me redraw that here. So from there, that picture to this picture here, you have, here's your large subunit, your small subunit, your three binding sites. Here's your mRNA. So now, if we look at everything shifting over, so this AUG is now in this site. This AGC is now in this site. And now the UAA is now in that third site. Now what is it? As it shifts, it brings with it its tRNA molecules too. So as that AUG shifts to here, it brings with that UAC tRNA molecule. But that tRNA no longer has MET attached to it because it broke the bond and now it's attached to that one. So this one's all by itself. This one is now shifted over here. So this UCG now has serine as well as methionine. So it has serine and MET. So it has the two amino acids, and this codon is now in that third spot with nothing there yet. Okay, so that's what it looks like. So now what's going to happen next? This tRNA has done its job. So therefore, guess what? The E site, this first site, stands for exit. So this tRNA molecule is going to exit. And this will be empty because it's brought its first amino acid. This tRNA can go be reused and go find another methionine in the cytoplasm and go find that and reattach to it. So you reuse these guys, so this can find another methionine and be reused. This is just hanging out here. This middle contains the growing amino acid chain, and then UAA would be the next codon. So you'd use your chart, UAA. UAA, when you use your chart, happens to be the stop codon. Does the stop codon code for amino acid? No. All right, so let's look at what happens with the stop codon. But before I do that, just a real quick, if this wasn't a stop codon, then what would happen? Another tRNA would bring the next amino acid. This bond would break, it'd reattach, and it would keep on growing like that until a stop codon is reached. When a stop codon is reached, there's no tRNA molecule that will bind to it. So what happens is that you get a release factor that binds to it. Now what happens when this release factor binds to it? It breaks this bond here. So therefore your amino acid chain, in this case I made it short just to make it short and sweet here. But in real life you'd have many amino acids long, so in real life this would be a polypeptide chain. This is really not a polypeptide because it doesn't have very many. But it would be a polypeptide chain that's broke off, and this would be your completed chain because we would be done. And then everything else dismantles. So what does that mean? The tRNA, this UCG, is now floating around by itself. All right, so the tRNA comes off. The mRNA detaches. So you have your mRNA detaching. Here's your mRNA. And then the ribosome breaks apart into the large and small subunits. So it'll stay broken apart. So this is the large, small subunit until it's reused for protein synthesis again. And so that's the termination part of proteins of translation. And so this whole process here, so that was transcription, RNA processing. This part is what? This was all translation. And that's protein synthesis in a nutshell. Is that helpful?
Yeah.
What happens if you use this versus stop codon?
This is your fly lab.
So
People
because
did really
it's a
well
stop
on this.
codon, that
As a whole.
makes it have a release factor. So release factor means
Um,
everything basically explodes.
I do have
So it just individually comes
a
off.
small little chi-square. I'm gonna give you some data and have you tell me whether or not it's
Right,
acceptable.
So see this?
So I do have that.
So this was attached to this, but it's
Oh.
broken up like this.
Oh, no.